Hello everyone, welcome back to the online lectures. This chapter 16, we will be focusing on the molecular basis of inheritance. So this means we're going to be focusing on DNA. We're going to cover the structure of DNA pretty thoroughly, and then we're going to look at the process of DNA replication. Of course, this happens during mitosis and meiosis. If you need a review on those chapters, look back at chapters 12 and 13. In this chapter, the objectives are that students will describe or model the regulation and expression of genetic information, particularly describe or model the structure of DNA and DNA synthesis, which we call replication, in eukaryotic organisms. Let's get a little bit of context for this chapter. We'll bring back Chapter 14, where we learned about Mendel and the gene idea. Remember, Gregor Mendel was an Austrian monk who performed various experimental crosses using plants, particularly pea plants. He was looking at different characters like pod color, pea color, and texture, flower color, and he was noticing these ratios. He had a 3:1 ratio with the monohybrid cross. We had a 9:3 to 3:1 ratio with the dihybrid crosses. And from this, he was concluding that there was a heritable factor that was being passed on from the parents to the offspring. But he really didn't know where this heritable factor was coming from. This was occurring in the mid to late 1800s, so our scientific tools weren't really up to par yet for figuring that out. But fast forward to the end of the 19th century, 1875, 1890s, when cytologists were trying to work out mitosis and meiosis, biologists were seeing parallels between the behavior that Mendel described with his hereditary factors and what they were observing with chromosomes. And then in 1902, Sutton and Boveri began to develop the chromosomal theory of inheritance. And then Thomas Morgan Hunt did his famous experiments using Drosophila melanogaster, the Drosophila fruit fly, where he noticed different traits for the eye color factor. So he saw the wild-type red-eyed flies, and then there would be a white-eyed mutant fly. But the mutant fly would only pop up with male fruit flies. So he found evidence for X-linked to sex-linked genes, which further supported the chromosomal theory of inheritance. Around the beginning of the 1900s, this is when we figured out that the DNA was our substance of inheritance. And this became then the most important, most celebrated molecule because it contains everything that makes us us. And we know that if you have damaged DNA, this is not good, right? This is what causes cancers and aging and other things. So, fast forward a little bit, the mid-20th century, so 1950s, Watson and Crick, James Watson and Francis Crick, are credited with introducing what the DNA actually looked like, the structure of it, because now we knew what it was doing, how it was functioning, how it replicated, but we still didn't know the shape. So they introduced the double helix model for the structure of DNA, which, make sure you know, stands for deoxyribonucleic acid. We now know that DNA directly programs the development of biochemical, anatomical, physiological, and to some extent behavioral traits. So of course, this is the most celebrated molecule of our time. By the way, here are Watson and Crick. You see that they've actually built a physical model of the DNA. You can see the two strands. Here's one strand and it's wrapping, because this is a helix, right? And here's the other strand and it's wrapping. And between them, we see that they are connected. So we're going to look at the structure. It's really important to know the structure of the DNA molecule. And here it is. Here is the DNA. We did cover nucleic acids and their structure in chapter 5. So if you would like to go look at that part of the chapter 5 video, that would be helpful. Like I said, it's really important to know the structure of the DNA molecule, but I'm going to go over it here as well. So first note that a DNA molecule is made up of two strands, right? Those two strands are what are twisting in this helix shape. And this is why it's called a double helix, because there are two strands forming this helix. The nitrogenous bases that are making up the DNA are A, T, C, and G. And there are pairings that only will match with themselves. So adenine will always pair with thymine. So A is always with T. And guanine will always pair with cytosine. So G is always with C. Now you'll notice over here, this is actually looking at the molecular structure, so we can see what's building this up. You can see that with C and G, there are 3 bonds that hold them together, and these are hydrogen bonds, whereas A and T carry 2 hydrogen bonds that hold them together. So, for example, if you were to have a DNA molecule that has a predominant amount of CG binding, it would take more energy to break them apart because there are more bonds holding them together. So if you had a molecule that has 80% CG and only 20% AT, that would require more energy than if a molecule only had 30% CG. All right, let's go back over the monomers. So nucleic acids are DNA and RNA. This is DNA. Nucleic acid is a polymer. This is a polynucleotide. The monomer, the units that make up these polymers, are called nucleotides. And there are 3 things that make up these nucleotide monomers. And make sure you know these. So the first thing, this is a nucleotide monomer. These are the 3 things. 1, 2, 3. The first thing is the nitrogenous base. So this nitrogenous base is A. In this nucleotide monomer, this nitrogenous base is T. In this nucleotide monomer, it's C. In this nucleotide monomer, it's G. So those are the nitrogenous bases. So that's the first thing, nitrogenous base. Second thing is a pentose sugar. This is a carbohydrate right here. This is a monosaccharide. And it's a pentose because this is a 5-ring sugar right here. And this pentose, because this is DNA, the pentose is deoxyribose. If this was RNA, the pentose would be ribose. And then the third thing is the phosphate group. So for DNA, we have the nitrogenous base, A, T, C, or G. We have the pentose sugar, which is deoxyribose, and we have a phosphate group. Now, this is the monomer, and they connect. The phosphate group of one connects to the pentose sugar of the other. So here's the second one. This is going to connect here on the third one, and this is connect here on the fourth one. This is the DNA backbone here. This is the blue ribbon that you see in this image. So this is called the backbone, and you might hear it called the sugar phosphate backbone because it's solely made of the pentose sugar and the phosphate. Sugar phosphate, sugar phosphate, sugar phosphate. So this is the sugar phosphate backbone. So how are these 2 strands, these 2 backbones held together in DNA? Well, that's where the nitrogenous bases come through. Again, they're held together by hydrogen bonds, and that's it. Note that all of this, the sugar phosphate backbone, the monomers here, those are all held together by covalent bonds. Okay? These are very strong bonds. They're not just going to break with a little bit of heat. They would need a specific enzyme to cleave this off. On the other hand, the hydrogen bonds break more easily. They'll separate with heat, or we'll see there's an enzyme that kind of comes through and just unzips them, which is important because we do need these to separate and anneal back together while we're duplicating our 2 strands of DNA, and we'll see that in this chapter. Okay, the other important thing about DNA is that it is anti-parallel. Anti-parallel. So what does anti-parallel mean? Well, we see that the two strands of DNA have directionality. One end is said to be a 3' end and the other is said to be a 5' end. They're anti-parallel because they run with the 5 to 3 in this direction and then the 5 to 3 is in this direction. So they're opposite. They're not parallel. If they were parallel, it would be 5 to 3 going both would be going in the same direction. But they're anti-parallel. So what does the 5' and 3' mean? This is important because it has to do with how the strand is able to build. If we want to elongate this strand, you can only add it onto one side. So let's go through how to count that real quick. Okay, so remember that this is the pentose sugar and this is deoxyribose. Ribose is a 5-carbon sugar. Sugars, monosaccharides tend to be cyclical like this in our body. You've also seen in previous chapters where we can draw out ribose in a linear form. Here's a slide from chapter 5 when we were looking at carbohydrates. Here are our sugars, just as a recap. Remember, monosaccharides are the monomers that build up the polymers for carbohydrate polymers. We call the monomers monosaccharides. We call the polymers polysaccharides. So glucose is an example of a monomer, which we have here. Fructose, which you all know is the sugar that's in fruits. Here's ribose. So ribose is a 5-carbon sugar. It has the molecular formula C5H10O5, and this is the linear shape. So imagine this carbon over here wrapping underneath to join with this oxygen. So that would form a 1, 2, 3, 4, 5 ring structure, and this part will be hanging off. That is how we form this pentose. So to count the carbons, we start at the oxygen and we go clockwise. So this is carbon 1, this is carbon 2, carbon 3, carbon 4, and here is that 5th carbon. So when we say 3' and 5', what we're talking about is at which carbon things are attached to. So this is carbon 1. So the nitrogenous base is always on the 1' carbon. Here's carbon 2. Here's carbon 3. So 3', if you look down here, carbon 1, carbon 2, carbon 3, the 3' end of this strand is, because this is the end of this strand, the 3' carbon. And underneath the 3' carbon, we always have a hydroxyl group coming off. This hydroxyl group at the end of the 3' carbon, think of it as like a sticking point, a sticky hydroxyl. Oxygens like electrons, we know they're very electronegative, so they're kind of like a vacuum for electrons, even if those electrons are attached to other molecules. So this is a good place for binding to occur. And we'll see that when we want to elongate a strand of DNA or RNA, we always have to do it at the 3' end, right at this oxygen. So looking up here at the 5' end, we have carbon 1, 2, 3, 4, 5. Off of this 5' carbon is the phosphate group. So the 5' end always has the phosphate group. I just remember that because they both have the same sound, right? 5', phosphate. That's just a quick and easy way to remember that. And then again, the 3' end, we have the hydroxyl group, which is where all of the binding is going to occur. Same thing here, 5' end, so the phosphate group's down here, and we go, it's also flipped upside down if you notice. This is, all the oxygens are up at the top here, and they're flipped upside down. So 5' end, and over here is the 3' end. So if we were to elongate this, we'd have to come here. If we're elongating this one, we have to come down here. Another important aspect about DNA is DNA complementary base pairing. What we're talking about is those nitrogenous bases, A, T, C, G. You now know that A binds with T and C binds with G. Because of this DNA complementarity, each of the strands acts as a template for building a new strand when we're going to replicate it. So as we're going to see, we have our two strands of our double helix held together by hydrogen bases, hydrogen bonds in between each of these nitrogenous bases. Now, during the DNA replication, that parent molecule, that DNA molecule, unwinds, and it also unzips. So these two strands come apart. This allows all of the cellular machinery, all your DNA polymerases, all your enzymes to get in here. These then become template strands. The cellular machinery reads them, and because of the complementary base pairing, it knows exactly which base to put on here while it's elongating the new strand. So the dark blue strands are the template strands for two new DNA molecules to be created. And here we have G as the template, the new complementary base is C, A is the template, the complementary base is T, so on and so forth. These new strands are referred to as daughter DNA molecules. The first one is the parental strand. So one parent molecule will create two completely identical daughter DNA molecules. We know that that is how replication works now, but when the scientists were working this out 50, 60 years ago, they really didn't know. So the correct model that we just walked through is called the semi-conservative model, right? So we have one parent strand, which we're showing in the dark blue, and they separate and each become a template for two daughter strands. So this is why they are semi-conservative. The new daughter strands that are created are half of that, right? Half of that. One of the strands is the same exact thing as the parent strand. So that's why it's half conservative, semi-conservative. They weren't sure if you were look at these other models, the conservative model, if this is the parent strand, they thought that out of the two new strands, one of them was just the same parent strand, and one of them was completely new molecule that was built. And in the dispersive model, they thought maybe there were somehow pieces that were cut off and replicated and then pieced back together so that the daughter strands were sort of a mix, a mosaic of both conservative, the parent molecule, and new molecules. So we know that neither A nor C is correct. The semi-conservative model is the one that is correct for DNA replication. As a reminder, DNA replication is occurring during interphase. So this is the chapter 12 PowerPoints. This is the cell cycle. We have interphase and the mitotic phase, or M phase. Interphase has three separate stages, G1, S, and G2. The S phase stands for the synthesis phase, particularly we're making DNA. This is DNA synthesis. So if the cell cycle is the cycle at which the cell is living, 90% of the time it's in interphase, and then for 10% of the time it goes through its mitotic phase where the genetic material organizes, it condenses, it organizes itself across the metaphase plate, and it separates into two new distinct cells. So of course, in order for this to happen, we need to duplicate the DNA. So that is what DNA replication is. It's DNA synthesis. We are going to look at DNA replication for both prokaryotes very briefly, just so you can see it. And then we're going to go more deeply into eukaryotic DNA replication. So we're making DNA, we're making a copy of the DNA, and it happens at very high speeds. We'll get some of those numbers as we go through the slides. We're going to be going over a list of about 10 or so enzymes that are really important for the process, and I'm going to walk through each one of them. They're very important for you to know. You have to know all 12 of them. And we'll cover the origins of replication, the spots along the chromosome where replication has to begin. It's like a little start site. Of course, eukaryotic chromosomes are linear, right? We have 46 chromosomes, and with the exception of the Y chromosome for males, all of the other chromosomes have thousands of genes, and so they have thousands of origins of replication as well. Your DNA molecules are quite large. I believe if you unravel all the DNA from one cell, it's about 6 feet long. So just think about that. And we'll look at the directions of replication as we're going through it. So here is replication, DNA replication in an E. coli cell. E. coli is, of course, bacteria. So this is a prokaryote. This is bacterial replication. Notice again, since it's a prokaryote, notice that the DNA is a circle. This is a plasmid. So let's look at the origin of replication. The origin of replication it's just what it sounds like. It's the place where DNA replication, that's its start site. It's going to have a unique code that's there that allows for other cellular machinery to jump on there so that all of the proper enzymes are in place. So the origin of replication, here we only have one of them. And what happens is, remember, these are two strands of DNA. They're held together by hydrogen bonding, and we need them to separate so that we can properly put the complementary base pairs onto it, right? So the origins of replication get separated, and all of the enzymes jump in here and start making the new DNA. The new DNA in this picture is the light blue strands, and we have one for each of the dark blue is the parent strands. So along each parent strand, which is acting as the template strand, we have a daughter strand that is being created. This forms like a little bubble here. So this is called the replication bubble. And at the edges of each bubble, we see that it's a fork. This is the replication fork. There are two forks, and replication is occurring in both directions, into both forks. We're going to see this again when we get into the eukaryotic, because it's very important. There's something very important about the directionality of how DNA is built. So for now, you can just see that the creation is into both sides of the forks here. And this bubble is going to get larger and larger, right? It started off small, it's getting larger as these two parent strands are separated. This is very large now, right? Here's our origins of replication. And we have created a large piece here of the daughter strand, and there's only a little bit more. It finally finishes, and then it just separates into our two new daughter DNA molecules. Again, you see the semi-conservative aspect of DNA replication, where this new molecule is half parental, half new daughter DNA. Same with this one. This over here is origins of replication in a eukaryotic cell. So here we see that there are multiple origins of replication. They're showing 3 here. Remember, there can be thousands along one chromosome. The same thing is going to be occurring where the DNA is opened up here, the hydrogen bonds are split, and the replication bubble along with the 2 replication forks are formed, and the new daughter DNA is going to be created into the forks on the top this way and on the bottom this way until the bubbles meet each other. So we can see they're expanding and expanding until finally we have 2 separate daughter DNA molecules. Okay, let's look at the actual process of DNA replication. So this is where paying attention to the names and the functions of the proteins and enzymes is really important. So in this image, we see a half of a replication bubble and one of the replication forks. So just remember, we're only looking at half of it here. The same exact thing is happening in the going the other way in the opposite direction, but on the opposite strand. So if we're on the top building this way on this side of the fork, on the other side of the fork we're going to be on the bottom building in to the right side replication fork. And we'll see why in a minute. So you've started at your origin of replication. The enzymes that are responsible for unzipping the two strands of DNA is called the helicase. So the helicases are enzymes that untwist the double helix at the replication fork. So there's going to be one at this replication fork, and there's also one opening up the other replication fork on the right side. As the helicase is unzipping the DNA, remember, all this is doing is separating the two nitrogenous bases, which have an affinity for each other. They want to bind. They want to reform that hydrogen bond. So in order to stop that from happening, to prevent that, we have these little single-stranded binding proteins that are there to kind of cap it off, to make sure that these strands don't anneal back together with each other. So single-stranded binding proteins. You sometimes might see that SS instead of single-stranded written out. You'll see this also with, we'll say double-stranded, DS. So SS, single-stranded binding proteins. If you ever see DS, it means double-stranded. Now remember, the DNA is two molecules which are twisted around each other. Imagine you had two strings that you twist around each other, and then you hold one end of them, shut so that nothing can uncoil, and you open the other end and you pull it, the two strands away from each other. At the very bottom where we have the closed end, the coils that were there would tighten. They would become supercoiled. And the same thing would happen with the two DNA strands. We have this helicase enzyme which is unzipping, and all of these coils would continuously supercoil. They'd become even more tightly coiled. And this is DNA, and we don't want to damage our DNA by overcoiling it. So, while the helicase is over here unzipping the DNA, topoisomerase is on the other side of this fork ahead of it, unwinding the DNA and it does this by cutting one strand of the DNA. It's just a single-stranded cut. Very important that it's not double-stranded, but it's a single-stranded cut, and then it basically unwinds it and then puts it back together. So for every, let's say, coil that this opens, this has to uncoil it on this side. Here's a representation of that happening. So we see how it's cut one strand and then it looped it around, unwound it, and ligated it, glued those two pieces that it cut back together. So topoisomerase is ahead of the helicase, and it corrects overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. Now, those three things, the helicase, the single-stranded binding proteins, and the topoisomerase, are all enzymes and proteins that help us prepare the DNA, but we haven't yet started to make it. So this next enzyme, primase, the pink one over here, primase's job, to prime something, it means to get it ready for something, right? So what primase does is it puts on a primer, an RNA primer. So we want to build DNA. We're doing DNA synthesis, but in order to get that going, first we have to put on an RNA primer. And this RNA primer is a short strand, it says here, 5 to 10 nucleotides long, not long at all. And this is because the enzyme that's responsible for elongating DNA, for building DNA, can't initiate synthesis of a polynucleotide, they can only add nucleotides to a 3' end. So we need something here first before our DNA builder can come in and start adding on. I know it's strange, but it's true. So primase adds on the short strand of the RNA primer. And remember, we can only build on the 3' end. The 3' end. Here is a review of that really quickly, right? We have our 5' end, which has the phosphate. We can't add anything onto this phosphate. On the 3' end over here, we have the sticky hydroxyl group. So we are constantly adding onto the 3' end, and this is for RNA and for DNA. So because we are adding on the 3' end, we build things in a 5' to 3' direction. 5' is at the head here, and 3' is the tail. So we build RNA and DNA in the 5' to 3' direction. Note that one for yourself. Now we can actually start synthesizing our new DNA strand. The enzyme that builds the DNA strand is called DNA polymerase. And if you look at the names of enzymes, they usually tell you what they do. A polymer, what's a polymer? It's a group of monomers put together, right? And this is a DNA polymerase. So anything that ends in ase is an enzyme. So this is just an enzyme that creates DNA polymers. So DNA polymerase. There are actually a few different types of DNA polymerase. We're going to see DNA polymerase 1 and 3. There's a DNA polymerase 2. There's a few different ones in prokaryotes versus eukaryotes. Just as a little heads up, if you're going into microbiome or in your future courses, if you see, pay attention to which DNA polymerase we're talking about. So, DNA polymerases catalyze the elongation of new DNA at a replication fork. What do they need? DNA polymerases require that primer, that RNA primer, put on by the primase, and of course they require a DNA template strand. The parent strand was acting as the template strand. We need the template strand because this is how we decide which complementary base pair, this is how the DNA polymerase decides which complementary base pair it's going to add. So as the example shown here, we build the new strands 5' to 3'. Here's that 3' OH there at the bottom. So this is where we're going to be adding on the complementary strand for A. Of course, A always binds with T. So this is our nucleoside. This is thymine. The DNA polymerase enzyme is going to pop this on underneath here. This is going to create a phosphodiester linkage, if you remember that from chapter 5. Phosphodiester linkage. This phosphate group is going to be binding onto this sugar over here, and then we're going to lose 2 of these inorganic phosphates here. The 2 of them together is called a pyrophosphate, and now we have elongated our strand, and it's going to continue. The next one would be a guanine. So this rate of elongation is 500 nucleotides per second in bacteria and 50 per second in humans. So it's a lot faster in bacteria, but still very fast, 50 per second for us. That was the explanation for the general process. Now we have anti-parallel elongation, meaning both of these parent strands, the top dark blue strand and the bottom dark blue strand, have to be elongated. But there's a little bit of a problem. So look at this picture up here. We were looking at this replication fork in the last couple slides over here. I'll bring it back over here, right? We were looking at that bit. But I told you there's a whole nother side to it here. The DNA polymerase enzyme can only build 5' to 3'. So if the origins of replication were here. We put our RNA primer, the red piece here, that's the RNA piece. The DNA polymerase would have jumped on there and began putting on the nucleotides at the 3' end. And in that way we build 5' to 3'. There's a problem because as you open up both of these strands and you start at the middle here, you'll go this way, but then you leave this whole part of the strand being unreplicated. We actually have two different ways that the DNA is replicated. We will have a leading strand, which is able to continuously just elongate that DNA. It's going to be going toward the replication fork. Nothing's stopping it. It's just continuously adding on the nucleotides onto the 3' end, like this right here. So here's our RNA primer. The light blue is the DNA. This is the DNA polymerase enzyme. And you'll notice this is DNA polymerase, which is abbreviated DNA pol 3. So DNA pol 3 is the elongator. We are elongating at the 3' end and we're moving into the replication fork. Now, of course, this helicase is going to continuously unzip this DNA molecule. The topoisomerase is in the front, continuously unwinding this DNA strand. The single-stranded binding proteins are here to make sure that this doesn't close. What's the problem over here? If we have the leading strand going toward the replication fork on this part of the top strand, this part of that top strand isn't getting replicated. So in order to elongate the other part of this strand, which is called the lagging strand, DNA polymerase has to work in a direction that goes away from the replication fork. So the leading was moving toward this replication fork, and the laggings are moving away from their side of the replication fork. So you're going to have to use your imagination for this one a little bit. Imagine that actually we can just use it as this is here. So these origins of replication and this replication bubble is continuously elongating. And as it does, it opens up new territory, new parts of this parent DNA strand. So imagine that this helicase unzips this a little bit more, and this is now able to be replicated. The leading strand for this strand here, because we have anti-parallel elongation, both the top and the bottom anti-parallel strands need to be replicated. So on this strand, again, this is moving toward the replication fork, and this is in the 5' to 3' direction. So this is our leading strand. But so as we open up this DNA, this is just going to continue to replicate this parent strand. The top strand over here is going to be elongating a small section away from the fork. So we're going to have a little RNA primer put on. The DNA polymerase is going to jump on, and it's going to move in this direction so that we're still moving in the 5' to 3' direction. If you notice, these arrows are all going in the same direction. They're all satisfying the 5' to 3' direction. Same with this one here. In this strand, the bottom strand, if the top strand is 5' to 3' this way, then the bottom strand is 5' to 3' this way. Now, however, you'll notice that these lagging strands, this lagging strand, they're creating pieces, fragments of DNA. These fragments have a special name called Okazaki fragments. Now we have a new enzyme called DNA ligase, which is going to come in and glue these pieces together. Here's another representation of the lagging strands. We are looking down here, right? So here's our origin of replication. Our leading strand is going toward the replication fork. our lagging strands are going to be going away from this replication fork. Here's our first Okazaki fragment that's being created. The primase puts on the RNA primer. Again, we always build 5' to 3'. After the primer is on, the RNA primer, DNA polymerase 3, DNA pol 3, that's this pink enzyme, was able to latch on and start elongating DNA strands by adding nucleotides at the 3' end. So we're building, this light blue is the DNA. It's building into the previous one that was there. So this one was built first. As this replication fork was opening, the second one was able to be built. Once it opened even more, a third one is built. Now it's opened even more. And as this continues to open, again, we're going to have a fourth one created here. We're going to have a fifth one created here, right? So we have these two fragments that are sitting here, and they're not connected. Of course, we need them to be connected. We have another enzyme called DNA pol 1. That's this yellow enzyme. DNA pol 3 is the pink one that's elongating. DNA pol 1 is removing the primers. We can't have RNA in our DNA strand. DNA polymerase 1 is removing the red RNA primer pieces. And then once the red primer pieces are removed and replaced with DNA, DNA ligase, this green enzyme, comes together and comes and glues these two together. It ligates them. This is the entire process. Let's just walk through it together one more time. So we have our origin of replication somewhere over here, which is where the replication process started. It opens up by breaking the hydrogen bonds to allow all of these enzymes to get in, and we create this replication bubble with two replication forks, one on each side. Both of these parent strands, the dark blue, they're anti-parallel. They're running in different prime directions. Both of them have to be replicated. Because DNA is only replicated in the 5' to 3' direction, the top strand is going to be elongated this way, while the bottom strand has to be elongated this way. The enzyme that unzips the helix, the two strands of the helix, is called helicase. That's this green enzyme, and there's going to be one elongating into the fork on each side. As it opens up the DNA, single-stranded binding proteins bind the DNA to make sure that it doesn't anneal back toward itself, because it very easily will. The RNA primer needs to be placed on to the parent template DNA because our DNA polymerase 3 elongator can't start out of nothing. It can only start by adding on to something that's already there. The RNA primase, the primase enzyme, puts on a short 5 to 10 nucleotide RNA primer. Then the DNA polymerase 3, DNA pol 3, can latch on to the primer and begin elongating it. Again, always 5' to 3' direction. The leading strand starts at the origin of replication and just goes toward the replication fork. Again, same with the bottom. This is going toward the other replication fork. The lagging strands, however, can't go from this way to that way. So instead, we have to create little fragments as this unzips, as the DNA opens itself up for all of these enzymes to get in. It creates these Okazaki fragments. The Okazaki fragments have to be glued together. So first, the RNA primers have to be removed. DNA polymerase 1 comes in and removes the RNA primers and replaces them with DNA fragments. Then the ligase enzyme will find any nicks, any openings in the DNA, and ligate them, close them up, so that we have just one continuous strand of DNA. Now, as we saw, DNA polymerase can elongate at a pretty quick rate. In bacteria, it elongates at 500 nucleotides per second, and in eukaryotes, it elongates at 50 nucleotides per second. Of course, there are going to be some errors that occur, and we do not want errors in our DNA sequence. Our DNA polymerase is able to proofread the newly made DNA, and in doing so, it can replace any incorrect nucleotides. As we saw before, DNA elongation occurs at a very rapid rate. In bacterial cells, it's 500 nucleotides per second. In eukaryotic cells, it's up to 50 nucleotides per second. And this can cause error. So luckily, we do have some proofreading mechanisms. The DNA polymerases themselves actually have a proofreading mechanism where they can distinguish between the template strand and the newly made DNA, and they can replace any incorrect nucleotides. They do this with mismatch repair. So remember, DNA is complementary. If you have an A in the parent in the template, you need to pair it with a T. If you have a C in the template, you need to pair it with a G. With mismatch repair, we're making sure that those are matched correctly, and the enzymes are able to correct any errors in the base pairing. We also have another form of repairing DNA called nucleotide excision repair. To excise something means to rip it out, to take it out, or to cut it out. Nucleotide excision is cutting out pieces of damaged DNA or incorrect DNA. We see this word nuclease. It's an enzyme because it ends in ase, and we see nuclei, right? That's nucleic acid. A nuclease is an enzyme that can cut nucleic acid. And of course, our nucleic acids are DNA and RNA. So like I said, enzymes usually tell you exactly what they do. So the nuclease is able to cut out pieces of DNA that has been damaged and replace it. Now we know that the DNA is probably the most important molecule in our body because it encodes everything that is required for us to live and to survive and to function properly. So of course it's never good when we have damaged DNA. And what are some things that can damage the DNA? Well, we know different chemicals can harm them. Physical agents such as cigarette smoke or different high radiation rays like X-rays or UV rays or gamma rays. If you ever see or hear the word mutagen, that is a substance that can mutate your DNA. But of course, not all mutations are bad. Mutations are the source of genetic variation for us. If it weren't for mutations, we wouldn't have all of these species that exist. So we know that we have proofreading repair for our DNA. It doesn't mean that it's error-proof. We, of course, have so many different mutagens. We have chemical mutagens, physical mutagens, and these sequence changes can be passed along from one generation to the next. If a mutation is favorable for whatever reason, that organism that inherited that mutation has an advantage, has a new advantage that the rest of its species do not. And that advantage will lead to that individual having a higher fitness, and that means having more offspring. And if they have more offspring, then they're going to pass on that new mutation. And that is evolution. That is the mechanism of evolution. It's mutations, these spontaneous, whether they're spontaneous or from missing something in error proofreading or from a physical or chemical mutagen, those mutations that are favorable are kept and passed down. And there we go. That is the source of the genetic variation upon which natural selection operates. The last bit about replication for this chapter has to do with replicating the very ends of our DNA strands. So this is only applying to DNA that is linear. So this has nothing to do with prokaryotes that have circular chromosomes. So what happens is you now know that you have the leading strand and the lagging strand. Well, with the lagging strand, you need to put on these RNA primers. So at the end of this strand, the one that's being elongated, that's 5' to 3', we're going to have to put the RNA primer on here at the 5' end so that we can get this piece of DNA here. Unfortunately, the replication machinery has no way to fill in this piece. We can remove the RNA primer, but remember, in order for DNA polymerase to elongate, it has to have something to latch onto. So if we remove this RNA primer, the DNA polymerase can latch onto these nucleotides and be able to elongate in this direction, 5' to 3'. We can't grab onto the 5' and elongate, well, this would be elongating in the 3' to 5' direction. We can't do that. We have to elongate 5' to 3'. So there's nothing for the machinery to latch onto over here in order to begin the process. So what happens is this is left incomplete, and so it actually gets cleaved off. And every single time the chromosomes are replicated, there are a couple of nucleotides, so anywhere from 5 to 10 nucleotide base pairs that are cleaved off and lost at the end of the chromosomes. And we do know that these are the telomeres. Telomere shortening is related to aging. Now, embryonic stem cells have an enzyme that is able to replenish the ends, the telomeres, and that enzyme is called telomerase. There are actually some cells that adult humans have as well. Sperm cells have telomerase, T cells, immune cells, epithelial cells, cells that require a lot of replication, where the chromosomes are constantly being replicated. Those cells will contain telomerase, but the vast majority of our somatic cells do not have telomerase. So again, that contributes to aging for us. One reason that's posited as to why telomeres shorten is that it might protect cells from cancerous growth by limiting the number of cell divisions that genetic material is able to go through. And interestingly enough, there's evidence of telomerase activity, that enzyme that's able to continuously replenish and elongate the telomeres in cancer cells, which may allow cancer cells to persist in living. Wrap up this chapter, let's finish off with looking at the chromosome structure, because it's important in understanding how the cell is able to wrap it up and condense it or open it up so that the DNA can be read for gene expression. Because remember, the DNA molecule is very long. I think it's the longest molecule in your body, each chromosome. And if you take all of the chromosomes in just one cell and you unravel it and put them together and measure it, it measures about 6 feet. So there's a lot of genetic material, big molecules here. So how is it that they fit so tightly into a cell? Well, it all has to do with how they're wrapped and coiled. So remember, bacterial chromosomes, those are prokaryotes. They have the double-stranded circular DNA molecules, and there's proteins associated with them that we're going to see. And then eukaryotic chromosomes, of course, have the long, linear, double-stranded DNA molecules with lots of protein. So what is this protein? The protein is called a histone. A histone has a tail on it. They associate into groups of 8, 4 on the top, 4 on the bottom, and each pack of histones like this gets wrapped with the DNA twice. So this is the double helix, right? Uncoiled. This is just, well, I mean, it's in its helical shape, but it gets wrapped around this twice. All along the DNA, all of our chromosomes get wrapped around the histones, and they form these nucleosomes. Anytime you see some, it's like a body. So this, nucleic acid body, nucleosome. So this happens along the entirety of the DNA strand. Then these nucleosomes get coiled. So they just imagine just wrapping, if you wrap these in a circle and you start coiling them. And this is a 30 nanometer fiber. And then that 30 nanometer fiber gets coiled and looped again. And this is a 300 nanometer fiber. And then those coils form into this chromosome that you think of. When you think of a chromosome, you usually think of it in this X shape. But this is you should know this by now if you already covered the mitosis and meiosis chapters. But of course, when we see that two of them together like this, these are two replicated sister chromatids. Pay attention to the words that are used to describe the different aspects of chromosomes. We have chromatin and chromatid and chromosome. Chromatin is DNA plus protein. So we mean DNA plus the histones. That's chromatin. Chromatid is the condensed version of the chromosome. If we have two chromatids that are replicated, remember those are sister chromatid. And this is called a replicated chromosome. So when we're talking about the genetic material during the cell cycle, the chromatin, which is DNA plus protein, undergoes packaging during the cell cycle so that it can be separated. Again, we want that to happen because the DNA is long and it will get entangled if it's not condensed. So the condensation helps protect it. However, during interphase, which takes up 90% of the cell cycle time, this is when the cell is going through all of the rest of its biochemical processes, whatever the cell is supposed to be doing for us. That's what it's doing during interphase. Here, the chromosomes won't be highly condensed. It says it'll be at the 10 nanometer fiber or the 30 nanometer fiber. So let's look back at this. Here's 30 nanometers and here's 10 nanometers. So pretty much during most of the cell's life cycle, it's not condensed. It's largely loose and open. And this has a special term that you should know. The loosely packed chromatin is called euchromatin. And euchromatin is the chromatin that would be able to be read for DNA replication or for transcription. If it's open, if it's loose, that means that we can get to it. However, even during interphase, there are some parts of the chromatin that are not loose. They're condensed, highly condensed, and we call the highly condensed portions of the chromatin heterochromatin. So it says here the centromeres and the telomeres. So the centromeres are holding the sister chromatids together, and the telomeres are the ends. So those would be condensed, and that's called heterochromatin. So those parts we wouldn't be able to read or do anything to, but they're not really necessary for this. So the takeaway from this slide is that chromatin can be either loosely packed or condensed. When it's loosely packed, it is called euchromatin, and this would be the regions that we can read. And then the densely packed regions are called heterochromatin. And this would be difficult for the cell to express any genetic information that's coded in these closed off condensed regions. Now, the cell is a highly fine-tuned mechanism, and it can open and close any parts of the chromatin whenever it needs to. It does this by altering the histones. It can it says, undergo chemical modifications. So what that means, if you recall, we talked about methylation in the last chapter, and when we learned the functional groups, you learned about the methyl group, CH3. Methylation is a chemical process where we just put a methyl group on this histone tail, and that is one of those. Or you can put an acetyl group, which is a little bit larger than the methyl group. The acetyl groups, since they're larger, there's steric hindrance where larger molecules can't interact too closely together. They take up more space, so you can't close them in as tightly. So that means that this chromatin won't be able to close in as tightly. Whereas methyl groups are much smaller, and they can help close the chromatin. The histones undergo these chemical modifications that can result in the chromatin either loosening or tightening. So that ends this chapter, chapter 16, the molecular basis of inheritance. You now know how to replicate DNA. Make sure that you know those enzymes and what they do. And in the next chapter, we will go into transcription and translation.